Wikipedia search will show that Robert Charles Gallo is an American biomedical researcher and is best known for his role in the discovery of the human immunodeficiency virus and in the development of the HIV blood test, and he has been a major contributor to subsequent HIV research. However, once secreted government documents revealed by Dr. Leonard Horowitz in his book, Emerging Viruses, AIDS and Ebola, Nature, Accident, or Intentional, show that Dr. Gallo has a direct communication to the creation of what we call the HIV virus. Now remember these names, people. Roy Ashe, Henry Kissinger, and Vice President Nelson Rockefeller were all financial investors in a company called Lytton Bionetics under the new National Cancer Institute Bionetics Administration at the Army's Fort Detrick facility. This is where AIDS was created. These people started a program called the Special Virus Cancer Program that began in 1962, which engaged in cancer virus studies using Lytton Bionetics supplied monkeys and chimpanzees. By 1966, Lytton Bionetics researchers advanced recombinant retroviruses causing the never-before-seen leukemia, lymphoma, sarcoma, immune-suppressive cancers, and by 1968, studies describing these developments were published. In 1968, Dr. Gallo was the National Cancer Institute's project officer at Lytton Bionetics, where new cancer viruses that were descriptively and functionally identical to HIV/AIDS were being created and tested. A year later, in 1969, Henry Kissinger, while serving as the White House National Security Advisor, selected the option for the use of these bioweapons for the military, and by 1970, the United States Defense Department ordered these viruses for use. Now we come to the German Merck family, which began selling drugs in 1668. Lytton Bionetics supplied the Merck pharmaceutical company with the bioweapon that we now call HIV and AIDS to the world, and this was even admitted by the company's then vaccine chief, Maurice Hilleman. And one of the first places they experimented these bioweapons on was mentally handicapped children at Willowbrook State School through a contaminated hepatitis B vaccination trial between 1970 and 1974. By 1976, after incubating the new Bionetics engineered and Merck supplied viruses for 2 to 5 years, the children at Willowbrook were rumored to be ill with a strange new disease. This was just the beginning of what we now call HIV and AIDS. This is the true story as to how HIV/AIDS was created during the late 1960s and early 1970s, originally stemming from Lytton Bionetics and distributed by Merck through contaminated vaccination trials. Now this is how AIDS was distributed into the American populace. This new bioweapon was then introduced to the American public through a hepatitis B vaccination trial conducted at the New York Blood Bank in 1978. The experiment at the New York Blood Bank was run by a man named Wolf Zimmunez, who joined the New York Blood Center in 1969 and headed its laboratory of epidemiology since 1973. This hepatitis B vaccination trial was specifically for gay men who were injected with the vaccine at the New York Blood Center in Manhattan during the period of November 1978 to October 1979. As quoted in the New York Times, Zimmunez was in charge of the hepatitis B vaccine field trials that began in 1978 and that were conducted among 1,083 male homosexuals. Researchers in these labs, led by Wolf Zimmunez, used the contaminated hepatitis B vaccines delivered from Merck, which infected these gay men in New York. Similar hepatitis B vaccine experiments were conducted in San Francisco, Los Angeles, Denver, St. Louis, and Chicago beginning in 1980. Months after the experiment began, The first cases of AIDS reported to the CDC appeared in young gay men in Manhattan in 1979. The first San Francisco AIDS case appeared in the city in September 1980, 6 months after the hepatitis B experiment started there. In June 1981, the AIDS epidemic became official.
Scientists at the National Centers for Disease Control in Atlanta today released the results of a study which shows that the lifestyle of some male homosexuals has triggered an epidemic of a rare form of cancer. Robert Bazell now in Atlanta.
Bobby Campbell of San Francisco and Billy Walker of New York both suffer from a mysterious, newly discovered disease which affects mostly homosexual men, but has also been found in heterosexual men and women. The condition severely weakens the body's ability to fight disease. Many victims get a rare form of cancer called Kaposi's sarcoma. Others get an infection known as pneumocystis pneumonia. Researchers know of 413 people who have contracted the condition in the past year. 1/3 have died and none have been cured.
Death didn't scare me. It was living with this for a long time. That's more frightening than death.
Investigators have examined the habits of homosexuals for clues.
I was in the fast lane at one time in terms of the way that I lived my life, and now I'm not.
The best guess is that some infectious agent is causing it. Today, researchers here at the National Centers for Disease Control said they had found several cases where people who had been sex partners both had the condition. The scientists say this probably means they are dealing with some new, deadly, sexually transmitted disease. The investigators see this as a serious public health problem.
From an epidemic point of view, there have been more deaths from Kaposi's sarcoma and pneumocystis pneumonia than have occurred with all the cases of toxic shock syndrome and the Philadelphia outbreak of Legionnaire's disease combined.
Researchers are now studying blood and other samples from the victims, trying to learn what is causing the disease. So far, they have had no luck. Robert Bazell, NBC News, Atlanta.
After the hepatitis B experiment ended, Wolf Zamunes insisted that all 13,000 blood specimens donated by gay men be retained at the blood center for future use. Several years later, when this blood was retested for the presence of HIV antibodies, government epidemiologists were able to detect the introduction and the spread of HIV into the gay community. Zamunes himself said that one day another disease may erupt and will need this material. The retesting of gay blood at the blood center proved that HIV was first introduced into the gay population of New York City sometime around 1978 to 1979, the same year the hepatitis B vaccine trials under Zamunes' leadership began. Similar to the outbreak of HIV/AIDS following the hepatitis B vaccination trials was the smallpox vaccine eradication programs sponsored by the World Health Organization in Africa. On May 11, 1987, the London Times, one of the world's most respected newspapers, published a front-page story entitled "Smallpox Vaccine Triggered AIDS Virus." The story suggests that AIDS in Africa is a direct outgrowth of the World Health Organization's smallpox eradication program. The Times story is one of the most important stories ever printed on the AIDS epidemic, yet the story was killed and never appeared in any major U.S. newspaper or magazine. Millions of Africans are reportedly infected with HIV. This large number could never have been infected by the simple act of a monkey virus jumping over to infect one African in the late 1970s. If that were the case, why don't we now have millions of AIDS cases in the U.S.? The obvious explanation for the result of millions of Africans infected with AIDS is just like that of the hepatitis B vaccines that were given to select gay populaces living in major metropolitan cities in the U.S. The vaccinations are biological weapons intended for mass depopulation. Henry Kissinger is quoted himself as stating in 1973, "Depopulation should be the highest priority of foreign policy towards the Third World, because the U.S. economy will require large and increasing amounts of minerals from abroad, especially from less developed countries. A decade before the first cases of AIDS, Dr. Donald M. MacArthur, a spokesman for the U.S. Department of Defense, told a congressional hearing that a supergerm could be developed as part of our experimental biowarfare program. This genetically engineered germ would be very different from any previous microbe known to mankind. The agent would be a highly effective killing agent because the immune system would be powerless against this super microbe. A transcript of the meeting on synthetic biological agents records the following comments of Dr. MacArthur. Number 1. All biological agents up to the present time are representatives of naturally occurring disease and thus are known by scientists throughout the world. They are easily available to qualified scientists for research, either for offensive or defensive purposes. Within the next 5 to 10 years, it would probably be possible to make a new infective microorganism which could differ in certain important aspects from any known disease causing organisms. Most important of these is that it might be refractory to the immunological and therapeutic processes upon which we depend to maintain our relative freedom from infectious disease. A research program to explore the feasibility of this could be complete in approximately 5 years at a total cost of $10 million. It would be very difficult to establish such a program. Molecular biology is a relatively new science. There are not many competent scientists in the field. Almost all are in university laboratories, and they are generally adequately supported from sources other than the Department of Defense. However, it was considered possible to initiate an adequate program through the National Academy of Sciences, National Research Council. The matter was discussed with the NASNRC, and tentative plans were made to initiate the program. However, decreasing funds in chemical biological research, growing criticism of the CB program, and our reluctance to involve the NASNRC in such a controversial endeavor have led us to postpone it for the past 2 years. It is a highly controversial issue, and there are many who believe such research should not be undertaken. Let it lead to yet another method of massive killing of large populations. Should an enemy develop it, there is little doubt that it is an important area of potential military technological inferiority in which there is no adequate research program. This testimony proves further that HIV was created in biowarfare laboratories organized by specific branches of the U.S. military and funded through U.S. Army government contracted pharmaceutical companies. During the 1970s, the U.S. Army's biowarfare program intensified, particularly in the area of DNA and gene splicing research. Renouncing germ warfare except for medical defensive research, President Richard Nixon in 1971 ordered that a major part of the Army's biowarfare research be transferred over to the National Cancer Institute, exactly where HIV would be discovered a decade later by Dr. Robert Gallo.